Hello, I'm Fernanda, talking from Brazil, and I'm going to present our study, Population Genomics, shed light on the origin, dynamics, and outcomes of a hybrid zone between two neotropical wildcats. Well, Leopardus is a genus of small wildcats distributed in the neotropical region of the globe. They have a complex evolutionary history with episodes of introgression and hybridization. And right here, where we have the Atlantic Forest and the biome Pampa meeting in southern Brazil, we have this contact zone between the distribution of a pair of leopard species that presents an active hybrid zone. Although they might meet also in another place of its distribution, this is the only known active hybrid zone, but we also have the occurrence of another leopard species, but only these two hybridize as far as we know. Well, these species are called Leopardus guttulus and Leopardus geoffroyi. They have diverged around 2 million years ago, and now they have this secondary contact, even showing very different habitats. Guttulus occurs northeastern to the hybrid zone, mainly in forested environments like this one, while the geoffroyi occurs southern to the hybrid zone, mainly in open areas of grasslands like these ones. And regarding phenotype, both species are also quite different in their coat pattern, their background color, their body length, and their body weight as well. And now here we have the habitat suitability maps present in red regions, the higher probability of occurrence of each parental species. And here, the same for the hybrids, right in the middle of both parental species. And is also where we have this intense modification of the landscape, mainly due to agricultural activities, suggesting that they have this tolerance of this type of alteration. Then, although these have been the targets of studies for a couple of decades, still there are several known aspects of this hybrid zone. Concerning conservation, the most relevant would be starting estimate the genomic structure of the related species, know how far this hybrid zone is reaching from the contact area, estimate for how long this is happening, and try to evaluate if it's a natural process or if it has been started by human activities. So trying to answer that, we have sampled a 4,000 transect within the distribution of both parental species and their contact area. We have sequenced at low depth around 200 genomes and plus 3 individuals at higher depth. This data passed through trimming and quality check steps. For mapping, we are using a Leopardus geoffroyi assembly at chromosome level as a reference. And since we are using mainly low-depth data, the base calling was performed paying attention to that, and the core of our downstream analysis were performed considering the genotype uncertainty of our data. And we have also built the complete mitochondrial genomes and performed phylogenetic reconstruction to investigate the matrilineal lineage history. And we also have performed clustering analysis on Y chromosome to evaluate the patrilineal lineage introgression. And we also have estimate global ancestry proportion and clustering analysis using the whole nuclear data set. And based on this, we identify our peer individuals, which were further considered to identify the ancestry informative markers, which were used for local ancestry inferences and time estimates of the hybrid zone. Here we have the overall genetic structure of our samples. The blue circles are the geoffroyi individuals and the red circles are the guttulus individuals. We can observe within the admixed individuals that we have a predominance of geoffroyi ancestry in them. And considering a threshold of 0.95, we have 40% of our individuals as hybrids. In the PCA, we observe two clusters of each parental species and the hybrids as a gradient between them. And when we look for the results in the map, we observe that the hybrids are mainly within the geoffroyi distribution in the Pampa biome, specifically in that region of this biome that we have more human activities. Regarding the evaluation of mating choice, we observe in the mitochondrial phylogeny that we have hybrids, here the colored individuals with both geoffroyi and guttulus mitochondrial genome, and we observed hybrids with both geoffroyi and guttulus Y chromosome as well. supporting that there is the contribution of both males and females from both species for the genetic pool of the hybrids. And finally, regarding the estimates of time since admixture using our local ancestry inferences, which is basically the posterior of a site in the hybrid genome that have been inherited from a parental or another, and the use of the length of these segments to estimate how old this admixture is. We've been observed that this current hybridization looks like to be happening for around 84 generations, which supports a very recent process of around only 200 years ago. And these results are consistent with the previous niche analysis that showed a higher density of hybrids in this outer environment compared to the parentals, also supporting that this process has an anthropogenic role. Furthermore, considering that the Pampa has this low rate of native vegetation protected and that have been observed in the last decades an increased transition into cropland in this area, this process could be quite concerning, especially for the geoffroyi in this hypothetical scenarios that the hybrid could expand more easily within a modified Pampa biome. So summarizing, we have the predominance of geoffroyi ancestry among the hybrids, as well their distribution within this modified Pampa biome. The hybridization looks like to be bidirectional, and the preliminary estimates of time suggest that this is a very recent process, and it may have started or at least intensified by human activities. So I'd like to thanks to the Biodiversity Genomics Organization and for the opportunity of talk a little bit about the project today. And I'd like to thank all the collaborators and people who is helping this project and the Brazilian funding agencies as well. And of course, thank you all for your attention.